Hi everyone, welcome back to video 4 of Unit 3. So what we just did was we went into this gross anatomy, this view from the naked eye. We talked about muscles and their different components, and then we went into the structure of gross anatomy. We separated these muscles versus fascicles versus myofibers, and we looked at the different connective tissues surrounding each of those. So now what we want to look at is we're going to look at this individual muscle cell, this individual myofiber. That's this guy right here. Individual muscle cell. And we're going to see that it's made up of these different things called myofibrils, that have these things called myofilaments, and we'll see some sliding filament. All of that stuff is what we're getting into now. So I'm going to write up a few different words that we want to become familiar with before we start talking about these muscles. Alright, so as we're getting into this microscopic anatomy of the skeletal muscle, when we're looking at cells, we're typically looking up under a microscope. So what cells are we looking at? We're looking at these muscle cells, these myo meaning muscle fibers. So these are single muscle cells. Whenever we learned about tissues, we talked about the 4 classes of tissues: epithelial, connective, muscle, and nervous. Very briefly, we mentioned when we're talking about muscles, we know muscles have this modified plasma membrane called a sarcolemma. So that's going to be our plasma membrane of the muscle cell. And then we also know that we have a modified cytoplasm, and that's called the sarcoplasm. So modified cytoplasm. And what are we going to find within the cytoplasm? If you remember in Unit 1 we talked about some cytoplasmic inclusions or some cellular inclusions. So these do have some cellular inclusions, and the skeletal muscles do have some cellular inclusions. One of those is myoglobin. So you've heard of hemoglobin before. That's that protein that carries oxygen around in the blood. Well, now we have a protein in the muscle cell that also holds oxygen. So muscles need oxygen and sugar. You need the sugar to get ATP. We'll talk about all that later on, but we need sugar and we need oxygen. So how do we store sugar? We have these glycogen. We store glycogen. So glycogen is it's a polymer made up of those monomers glucose. So this is our sugar, and we break this down to get ATP. And these two things, we'll call these cellular inclusions, cytoplasmic inclusions, sarcoplasmic inclusions, whatever you want to say. Alright, so that's our sarcoplasm. We know nuclei whenever we did the classifications of muscle tissue. We know if we're looking at skeletal muscle, we know that these are multinucleated because the smaller skeletal muscles fuse during development. So we'll put multinucleate. And that's because the fusion of cells during development. Okay, and then if you guys remember, we talked about this thing making up the cytoplasm. The cytoplasm had different things in it. One of it was that liquid cytosol, but then we also talked about this thing, the cytoskeleton. So the cytoskeleton are these different filaments. So if you remember words like microtubules and intermediate filaments, and what was the other one? Microtubules, microfilaments, intermediate filaments. Anyways, we're going to get some modification of those here, and that's going to give us our myofibrils. So these myofibrils are these modifications of the myofibril. Modified cytoskeleton. And the myofibril is the actual thing that's going to be doing our contracting. This myofibril can then be broken up into smaller things called sarcomeres, which we'll get into in a little bit. I'm going to leave this as myofibril for now. But when we look at the picture I drew back here, hold on. When we were looking at this, we know that each skeletal muscle cell has hundreds or thousands of myofibrils. So this is one myofibril. So if I was really drawing a single skeletal muscle cell, remember they're nice and long. So if I was drawing this like this, I would have this would be one myofibril, that would be a myofibril, that would be a myofibril, and then they're broken up into these smaller units called sarcomeres, and we'll look at the anatomy of a sarcomere in a little bit. But just so that you can see, this, I know I drew one back here. This was one myofibril in this skeletal muscle, but this skeletal muscle has hundreds of myofibrils within it. So myofiber has myofibrils. Okay, let's get rid of all this and keep up with some more anatomy terms. So we had myofibril. The next thing that we want to talk about is the sarcoplasmic reticulum. So this is another modification of an organelle that we've already seen. I'll let you take a guess while I'm writing it. So sarcoplasmic reticulum, this is a modified endoplasmic reticulum, and more specifically, we know we have smooth versus rough ER, so this is smooth ER. It's modified smooth ER. What's its function here? This is going to store calcium, which is important for muscle contraction. That's Ca2+, that's a calcium ion. I'll abbreviate calcium like that a lot. Excuse me. Let me draw a picture of a myofibril. So here's this myofibril made up of these different sarcomeres, like this. And I'll do the sarcoplasmic reticulum as purple. So let's say that this is my sarcolemma, that outer boundary, that plasma membrane. Within that, in this sarcoplasm, I have these myofibrils, and they're surrounded in this sarcoplasmic reticulum. So it just goes longitudinally like that. And then at certain points, we have these intersections of sarcoplasmic reticulum that are running perpendicularly. So I'll just scatter them throughout. And these intersections come in pairs. We have something in between those pairs. So let's identify what we have. Let's stick with the purple since we're still on sarcoplasmic reticulum. So sarcoplasmic reticulum is storing this calcium, and these perpendicular runnings, we'll abbreviate them TC for now. This is terminal cisternae. So a cistern is a tunnel. I think they're underneath France or Italy, something. There's like a tunnel system, and they call them the cisterns. So cisternae. And then terminal is your end point. So as you're going along the sarcoplasmic reticulum, you end at a terminal cisternae there. And then in between those are something called a T-tubule. So T-tubule. And all this is an extension, an inward projection of our sarcolemma. So you can see up top, you can see the sarcolemma, and you can see it indents like this. So it makes this hollow tube in there. Okay. So that's our T-tubule. I want to see if there's anything else we need. I think that's it for now. Oh, and then these things, if you combine a two terminal cisternae and a T-tubule in the middle, that's called a triad. So when we get into the physiology behind all these things, we want to understand the words that we're using. So these are all new words. Myofiber, it's a single muscle cell. It's modified plasma membranes called sarcolemma. The sarcoplasm, the cytoplasm of the muscle cell, it has these cytoplasmic inclusions, myoglobin, which is this pigment that holds oxygen. Muscle is storing its own oxygen for muscle contraction. We'll talk about that later, and we'll see why glucose is important. It's how we get our ATP. We'll talk about why ATP is important as well. The nuclei, we know that these are multinucleate from the fusion of cells during development, and now we're going into these other parts of cells like this modified cytoskeleton called the myofibril. There's hundreds or thousands of these in one muscle cell. And it's surrounded by this modification of the smooth endoplasmic reticulum, and that's called the sarcoplasmic reticulum. We'll get storage of calcium there, and at certain points of the sarcoplasmic reticulum, we get this perpendicular running, and those are called terminal cisternae. They come in these pairs with something in the middle called a T-tubule, which is an inward projection of the sarcolemma. Alright, looking at this myofibril, let's break it up a little bit more. We know myofibrils have these things called sarcomeres. So a sarcomere is from one of these lines to another one of those lines, and we'll get into some more specific names in a minute. But this is what's contracting. We're getting this pulling of these myofilaments, these modified cytoskeleton proteins. We're getting them pulled inwards, and that's what's causing this contraction. So you'll see if this was this long and it contracts, and we have things like this now. This is the shortening that we talk about when we contract a muscle. So it shortens and thickens. We get this thickening because we get this overlap of our thick and thin myofilaments, which we'll get into, like I said, in just a second. So sarcomeres, they're going to have two types of myofilaments, these protein filaments. You have a thick and thin myofilament. Your thick myofilaments are made of myosin, and your thin myofilaments are made of mostly actin. So I know that was a lot. Let's put some more words out here. And also, let me draw you another mental map. So we've worked through all these. We worked through the whole organ, this muscle, and then this bundle of muscle cells called a fascicle going into a single muscle cell called a myofiber. Now we're looking at these structural components, this modified cytoskeleton called a myofibril. We know that they're composed of multiple sarcomeres, and these sarcomeres are composed of these myofilaments, both thick and thin. Our thick myofilament is called myosin, and our thin myofilament is mostly made of actin. So now I'm going to draw a sarcomere, and we're going to keep working through this stuff. Alright, cool. So we are looking at one sarcomere right now, and myofibrils are made up of hundreds of these. So let's use dark gray to label some stuff. The first thing that we want to label are the ends of these sarcomeres. These are called Z lines. We can also call them Z discs. Either one. We know that we have these thin myofilaments, and these are mostly made of actin. We have these thick myofilaments. These are made of myosin. We have the purple over there. That's going to be... this is an elastic myofilament. We don't talk about it too much, but it's what's connecting these thick myofilaments to the Z lines or Z discs. This is called titin. And I don't think he said it this year, but he said it in past years. The protein that makes up the Z line is called nebulin. So whenever you get a nebulizing treatment for an asthmatic attack, I think it's relaxing this nebulin. Okay, is there anything else that we want to add before? Okay, so talking about these different regions of this sarcomere, and I do certain colors for specific reasons, so I'll keep these colors consistent throughout this lecture. So if you're following along with color, I try to do the same colors I do. If not, create a system that works for you and stick to it. And I'll get rid of this. And this. Alright, so we can do this one of two ways, and we'll do both. You can just decide what works for you. You have to find the I band. You have to find the A band. And you have to find the H zone. The other ways that you can do this, you can say that you need to find where you only have thin myofilaments, where you only thin. Where do you only have thick myofilaments? And then where do you have all the thick? which means that you have all thick and some thin. So let's try that. Let's divide them up before we try to find them. So first thing I'm going to do is this only thin. So if I'm my only thin, I need to go this end of the thick myofilament, which is right here, and let's start dividing that. So from the Z line to this point, this first dashed line, that's where we're showing only thin myofilaments. I know that you're seeing this elastin, but let's just pretend it doesn't exist. Or this titin. Let's pretend that it doesn't exist for now. Because right now we're only looking at thick and thin myofilaments. So, if you only have thin, this is your actin. This is your I band. How do you remember that? Once we get into this idea of troponin, tropomyosin, if that word overwhelms you, don't worry, we'll get to it. We'll come back and see how that helps you remember that this is only the thin. Okay, if we are going to do only thick, let's identify that. So only thick. So let's go to the end of my thin and draw a line down, because that would be only thick, and a line down, only thick. Well, if you notice, if I do that line as solid and this line as solid, what letter does this make? Maybe it'll be better if I do it in a different color, like yellow. So this and this. And I go across. What letter does that make? That's your H zone. So your only thick myofilaments is H, your only thin is I, and what else does that leave? Let's do a different color. Let's do this green. If I'm going to do all my thick, then I need to go from here, the end of my thick, and I need to go over here, which is the end of my thick. This is called your A band. So I'm going to redraw this, and I'm going to start talking, or I'll talk through it as I'm drawing it. So the first thing I want to know are the ends of these sarcomeres, called Z lines or Z discs. I know that I have thin microfilaments directly attached, and this is predominantly made up of the protein actin. I have these thick myofilaments here that are indirectly attached with these titin elastic myofilaments. These thick myofilaments are made up of myosin. And if I want to separate some different things, then I can separate 3 different regions. I can do only the thin, so I go to where my thick starts, and then I'm going to go from there over to my Z line. If I only have thin, that's I. Then I need to do only thick, so that's where I took my yellow marker, and I did only thick. So this was thick, and this was thick. And I said this letter made the letter H, so this is my H zone. Then I know I needed to do all thick. So if I did all thick, let's do this. That'd be from, I need a different color. If I did from here, that's my thick, and that's thick. This is the A band. So we've got Z lines, we've got I bands, A bands, H zone, and then if you go straight through the middle, you can do an M line. So we can say this M line is in between the 2 Z discs. We can say it's in the middle of the A band. We can say it's in the middle of the H zone. All those things mean the same thing. Alright, so when the muscle contracts, we're going to get these thick myosin filaments pulling our thin myosin filament, or thin actin. We're just going to say thick and thin. We're going to get our thick myofilaments pulling our thin myofilaments, so these Z lines move from the Z line towards the M line. So we contract inwards. Alright, now we want to go into the structure of this myosin and actin. As you can see, we've gotten smaller and smaller through these pictures. So back here, we're looking at the whole muscle. Then we went into one muscle cell. So when we went into one muscle cell, we saw hundreds or thousands of these different myofibrils. And now we're looking at one specific part of the myofibril, or one individual sarcomere, and now we're breaking that up. And now we're going even deeper into the sarcomere, and we're going to look at the anatomy of this stuff. So we're getting really small here. So we look at myosin. Myosin is a protein with a tail plus 2 heads. So what does that look like? Here's a tail. And if I have 2 heads, here's Here's a head, and here's a head. Each of these heads has 2 binding sites. So let's draw those. There's a binding site, and there's one. Okay. So that's a binding site. That's a binding site. There's one. And there's one. Now, what are these binding sites for? We have 2 binding sites, so let's figure out what they're for. One is for actin. So we need this interaction of myosin and actin. They need to bind together so we can get the pulling of the thin myofilaments from towards the M line. And then the other binding site is for ATP, and we'll define why those are important in a little bit. But if I look back at this thick myofilament, let's see what I'm erasing. All that. If I look at this thick myofilament, I have tons of tails. So these are all tails going in all different directions. And each one of these tails has 2 heads. So a head, for that. A head, head, heads. So all these different myosin proteins have all these different heads that are ready to grip and grab onto these thin myofilaments. Alright, so that is our myosin chain. So we have tons of myosin chains or myosin proteins, all with one. Each of these myosin proteins has a tail with 2 heads, and we see these 2 active sites. All we're doing right now is going over anatomy. We haven't done any physiology. We haven't talked about how these things work yet. So I want to do the thin myofilament next, and then we can talk about how these things interact to get muscle contraction. So we do thin. Do you remember what what protein makes up the majority of our thin myofilament? That's our protein actin. So here's our actin. And actin has a binding site for myosin. So this is the myosin binding site right there. But it's not always exposed. So we have this thing called the troponin tropomyosin complex. That's going to be covering this thing typically. So what is that made up of? I'll write it all out. So keeping up with colors, we have actin. We've got tropomyosin, which is this thread-like protein that wraps around actin. So we got tropomyosin. And then we have troponin, and troponin has 3 different parts. So we have troponin, let's do, since we're purple, let's keep troponin T. So there's troponin T. Next we have, let me erase this. Next we have troponin I. And then we also have troponin C. So this whole tropomyosin troponin complex, this is blocking our myosin binding site. So we have this head over here with this actin binding site just waiting to grab onto this actin, but it can't because it's blocked. A really quick note about this myosin, it has 2 configurations, basically 2 positions this head can be in. This is called our high energy configuration when it's shaped like this because it's ready to grab this actin. Once it reaches up, grabs this actin and pulls it towards the M line, then it will look like this. So the low energy configuration would be this shape. So these are the 2 heads in low energy, so high to low. And while this myosin is in its high energy configuration, it wants to bind to actin, but it can't bind to actin because on this thin myofilament we have this troponin tropomyosin complex. So that word is troponin tropomyosin complex. Again, we're setting the stage for all the anatomy to be able to figure out the physiology behind it. How do we get to this? How do we open up this binding site? How do we open up this binding site so that this myosin can grab onto actin and pull it towards the M line? What we need to do is we need to bind calcium. Calcium is going to bind to troponin C, so I'll have it bind to this red part. So when calcium binds to troponin C, troponin I lets go of actin. So we have this unit down here, this thing. So let me draw. I'm going to draw it when it's blocked and then when it's open. So when it's blocked, I have my troponin tropomyosin complex on it with my 3 different parts. So I have that. I've got troponin I, and I've got calcium. So when I put a calcium ion in there, what happens is this whole thing just kind of slides over. So this would be over here. Boom. And remember, this has the calcium bound to it. And now my active site is open. And then my myosin head can come up and grab it. So the whole point of this is calcium is the thing that needs to bind to this troponin tropomyosin complex to open up this active site so that myosin can reach up and grab the actin and pull it together. So now that we've set the stage, I'm going to end this video. I would highly recommend going back, watching this video again, put it on 2x speed, and listen to all this anatomy again. Because now we're moving into this physiology, these different processes, and you want to have a good idea from this microscopic anatomy. You want to be able to talk about all these different words: myofiber, sarcolemma, sarcoplasm, the nuclei, myofibril, sarcoplasmic reticulum, terminal cisternae, triad, T-tubule, myofilament, sarcomere, myofilaments, thick and thin, actin, this whole orientation of the sarcomere. You want all this stuff. Binding sites, troponin tropomyosin complex. You want to get all this stuff down before you start the physiology. Okay, I'm going to end this video and we'll move into the sliding filament theory of contraction.